I'm back. So I had to steal a iPad in order to finish this out. You know, last week. I'm just trying to survive this. So it's a little bit different than my normal setup, but we'll try and make it work. We're trying to get through oxidative phosphorylation. Um, we've kind of already started it, but we'll go through. And I'll remind you, we started with a molecule of glucose, C6H12O6. We then broke that into two molecules of pyruvate, which is has three carbons each. And then we broke down that pyruvate first to acetyl-CoA. I feel like I spelled that wrong. Um, and then we went through the citric acid cycle to completely break that down to carbon dioxide. So the result of the first two steps were we have a total out of all of this of 4 ATP, which isn't a lot, but we have a lot of reduced electron carriers. I think in total 10 NADH and 2 FADH2. And as I've said on numerous times, there's a lot of energy here in these electron carriers. And so now we're going to get at it via oxidative phosphorylation. So this is the third step in cellular respiration, and it uses the electron transport chain in which we take these high energy electrons and use them to build a potential energy via a proton gradient across the inner membrane of the mitochondria, and then that proton gradient is used to drive ATP synthesis from ADP and inorganic phosphate. And so this is a basic look. This is the mitochondria. It has an outer membrane here and an inner membrane. There are projections in the inner membrane that are called cristae, and that increases the surface area that allows for more reactions to occur. So the inner membrane is not permeable to ions. Particularly H plus, but any ions. The outer membrane is fully permeable because it has very large holes in it. So the outer membrane and the intermembrane space are basically the same. The cytosol and the inner membrane space, there's not much difference because there's basically free flow between them, but the inner membrane is locked away. The processes of the citric acid cycle occur in the matrix of the mitochondria, which is the space within the inner membrane. So everything that we've done thus far has been, outside of glycolysis has occurred within the matrix of the mitochondria. And the electron transport chain is on the surface of the inner membrane. And so this is the electron transport chain. So here, this is, shoot, this is where I don't understand what I'm doing. This is the matrix here and the intermembrane space there. And we're going to use the energy of the electrons from NADH and FADH2 that we generated previously to drive proton flow away from the matrix into the intermembrane space. So this is going to have a low concentration here and a high concentration, relatively speaking, there. This is a difference in potential energy, and that concentration gradient will be used to drive the protection of ATP from ADP and inorganic phosphate. Now, at the end of this, at the end of the electron transport chain, there's this electron that has to be dealt with. This is where the oxygen comes in place. So remember, oxygen is required for every step outside of glycolysis but the only time we use it is at the end of the electron transport chain. And what it does, this low energy electron picks up this oxygen and a couple of protons to form water. And so the only role for oxygen is to deal with the electrons that have been kind of used via the electron transport chain. And that forms water, and so water is a waste product of this process. And so here in electron transport, let's see, it's on the inner membrane of mitochondria. It has four complexes, 1, 2, 3, and 4. It also uses cytochrome C as an electron carrier and ubiquinone as an electron carrier. And both of these can hide those charged electrons and um, uh, transport them across the membrane of the mitochondria. And so that's shown here. This is complex 1. 2 is here, so 2 is not transmembrane. 3 and 4, and then the electrons will flow in via NADH in complex 1 and FADH2 in complex 2. And then with that flow, protons will flow through into the intermembrane space in complexes 1, 3, and 4. Since complex 2 is a peripheral protein, it's not transmembrane, it can't directly pump protons through the intermembrane space. This is ubiquinone here, and so the electrons come from complex 1 and complex 2 independently, and then go to ubiquinone, which transports into complex 3. And this is cytochrome C over here, and then things will come from complex 3 to cytochrome C, and over which will transport it to complex 4. And then the electron here in complex 4 links up with O2 to form water, obviously with some H plus in the process. And so here we go, NADH gets oxidized to NAD plus. The electrons flow to complex 1 to ubiquinone. Separately, FADH2 gets oxidized to FAD, and those electrons flow to ubiquinone, and the ubiquinone will carry both of those independently to complex 3. Um, and then I don't know that we'll get into it, but there's this kind of really neat thing called Q cycle that happens in complex 3, in which the electrons will go one at a time to cytochrome C. So there's a little kind of issue here. You have pairs of electrons coming from ubiquinone. Cytochrome C can only take one electron at a time, and so we have to deal with that other electron. And it kind of bounces around in complex 3 for a little while until cytochrome C is able to come back and pick it up. And that's called the Q cycle. Pretty neat. In the process, protons get pumped. So cytochrome C takes those electrons one at a time, and then by this time the electron is a fairly low energy. It links up with oxygen to form where was I? To form water at the end of the electron transport chain. And so remember the waste products of this whole process of cellular respiration. You go C6H12O6 plus O2 goes to form CO2 plus water plus energy, right? So the CO2 is formed in pyruvate oxidation and um, in the citric acid cycle, and the water is formed in the electron transport chain and oxidative phosphorylation. And here you can see the Gibbs free energy. And as we kind of alluded to earlier, these electrons, as they go through these pumps, the energy of those electrons are used to pump protons across the inner membrane. And so these electrons are progressively losing energy. So when they get through the end of complex 4, their energy levels is relatively low. And so they don't have the energy to pump protons anymore, but they pick up that oxygen um, and get reduced. So, because the pumps are using energy in order to drive something against the concentration gradient, this is an example of active transport, and we call those pumps. In this case, since they're pumping protons, we call them proton pumps. And yet another look at this with the protons. And so here you can see complex 1, complex 3, complex 4, all pump this. Complex 2 is interesting. This is succinate dehydrogenase complex. And this is directly from the citric acid cycle. And so we remember there is succinate dehydrogenase in the citric acid cycle that produces FADH2. That is this complex. It's part of the electron transport chain as well. And those electrons go directly into the chain. Remember, FADH2 is a prosthetic group, so it can't float off like NADH. And so the electrons have to be dealt with. In pyruvate oxidation, they were dealt with by transferring them in ADH. Here, they just throw those electrons directly into it. And remember here, succinate is the substrate. So a lot of times when we're trying to replicate the electron transport chain, we'll just add succinate to it, and it'll feed it directly into complex 2, which will supply those electrons to the electron transport chain. Now that we've built this gradient, then there's potential energy here. Oops, spelling's bad. And we can use that energy with the downward flow of these protons. So these are protons going back into the matrix. And we can use that energy to drive the production of ATP from ADP via the enzyme ATP synthase. And this is a gorgeous enzyme. It has a transmembrane unit called the F1, F2, F0, and then the catalytic unit called F1. And the way they work, it's taking the energy, the potential energy of this membrane, it is converting it to mechanical energy via this axis, and then translating that mechanical energy to chemical energy via the formation of ATP. We'll see that in a little bit more detail later on. So it's not quite as smooth as my other app here, but come on, move. There we go. Okay, so here are the names of the complexes, and you can tell by the names what they do. I said before, complex 2 is succinate dehydrogenase. Complex 1, NADH, ubiquinone, oxidoreductase. So what's going on here? NADH, which is reduced, is then passing on its electrons to ubiquinone, right? The same with complex 3 here, ubiquinone, cytochrome C, oxidoreductase. Ubiquinone, which is now fully reduced, is passing on its electrons to cytochrome C. Complex 4, cytochrome oxidase, and then ATP synthase. And this is a look of the structures here and then what's going on. NADH supplies 2 electrons, and it uses the energy of those electrons to pump 4 protons into the inner membrane space. They pass this on to ubiquinone, which is over here. At the same time, complex 2, FADH, is applying 2 electrons to complex 2. It'll pass those on to ubiquinone as well. Now, ubiquinone takes those electrons and it pumps 4 protons into the inner membrane space for these pair of electrons. and then passes those electrons one at a time to cytochrome C. Cytochrome C will pass it on to complex 4, where two protons get pumped into the inner membrane space. And so at the same time, this electron now gets linked up with oxygen to form a water. And so here, for each of these cycles, you have 10 protons being pumped from the matrix into the inner membrane space. And now coming back through, it takes 3 protons to convert to make a molecule of ATP from ADP and inorganic phosphate. And so you can see from each of these uh, um, electrons, pair of electrons, you can get, 10, 3, roughly 3 ATP from these electrons that are donated. And so this is where all the energy kind of comes in. And this is a look at the actual structures here. We don't have the crystal structure solved for complex 1 yet, but we think we know what's going on here. Well, actually, we don't know how these protons are pumped, but we think it looks like complex 3. And so here you see again, electrons go through, protons are pumped, they flow to ubiquinone. Complex 2 also takes its electrons and passes them to ubiquinone, and then ubiquinone passes them on to complex 3. Complex 3 will take those electrons one at a time and pass them to complex 2, and it pumps 4 protons as it does so, and then the electrons from complex 2 will pump 2 protons across. So I think, let me see if there's anything good. Anything else I'll say will belabor the point here. Um, One thing that's kind of neat, depending on how morbid you are, is that a lot of our poisons work on the electron transport chain. It's a kind of fundamental energy chain. So things like cyanide, carbon monoxide, they block the transfer of cytochrome C to complex 4. I don't know, oligomycin blocks ATP synthase, which shuts everything down. So a lot of these poisons permanently block these kind of systems in the electron transport chain and causes um, obviously problems with the ability to process energy. I'm not going to get into the voltage potential here, but there is, because we're talking about the transfer of protons, there is some change in voltage that occurs there. Uh, we've talked already about FADH2. I will show you the structure of ubiquinone, and it has 3 states, similar to FADH2. It has a fully oxidized state, has a semiquinone state, and a fully reduced state, right? And so here are where those electrons are kind of hidden in the reduced state versus the oxidized state. The thing is, it has this long hydrophobic tail, or sorry, hydrocarbon tail, right? So it's basically a lipid, and it's a lipid regardless of whether it's picked up these 2 negative charges or not, right? And we know that lipids can pass freely through a plasma membrane, and so ubiquinone is able to hide these electrons and travel at will to the through the inner membrane and mitochondria to get into the to go to complex 3. It's complex 2. I'm not going to talk about the Q cycle, but it's really fascinating, how this works and what's going on with the electrons. But this is a little bit complicated for an online lecture. I would do this if we were, IRL, but, you know, COVID and all of that. All right. And then we get to complex 4. So that looks like This is kind of critical. You know, one of my favorite test questions here is why protons, right? So we can form a concentration gradient with anything, but using protons is particularly genius for several reasons. So one, the delta G that's formed from this membrane potential is two things, right? It's a difference between this intermembrane space up here, the proton concentration, yes, so just a difference between the concentration, right, on one side versus the other. And so that's seen by this expression, right? Here, delta G is a function of the difference of the concentration. But because we're using protons, right, this is a charge. And so protons have a positive charge. By taking out the positive charges from the matrix and pulling them over to intermembrane space, you're causing a charge imbalance. And using protons in particular is really kind of genius. I mean, we could have done this with sodium or something like that. But what do we know from our studies of acid-base chemistry, right? We know that the proton concentration times the hydroxide concentration is always equal 10 to the minus 14, right? So what that means is that whenever you remove a proton here, you increase your hydroxide concentration, right? So by pumping protons across the inner membrane, you now create a lot of hydroxides in the mitochondria matrix, right? So you're changing the pH, and this then increases the membrane potential even more, right? So you have a lot of bang for your buck by using protons versus any other ion there. And so this is shown here. This is an electrical constant, this is Faraday's constant, another constant. The psi here is the difference in the electrical charge. And so you're getting not only energy from difference of concentration, but you're also getting electrical energy as well, which is adding to the delta G that allows you to make more ATP from that. We won't talk about that. Oops. Just stop it. Why are you doing this? There we go. So this is what I said. So that ATP synthesis, not only do you get the chemical uh, potential different in pH, but you also get the electrical potential based on the charge distribution on either side of it. And then looking at ATP synthase, um, I'm not going to go into too much detail, but it uses those flow of protons back into the matrix where the delta G favors it in order to drive ATP production. And I would refer you to this animation because it's pretty neat how this works. This is uh, a trimer here, each of what holds a ATP molecule and processes it. Let me see, but it's hard to see. I'll have to show you the video in class actually. Do that. Ah, shoot, on Monday. Come on. It's much more touchy than uh, my program. But here to see, this is gamma, so this rotates as the flow of uh, protons go through F0 here. And then if you look, it has basically three orientations. One is binding ADP, an inorganic phosphate. And then you have another, oops, it's probably this one, another that is having ATP bound, and then another one's empty, the release of ATP. And so as gamma, I've made a mess of this. And so here, gamma is in this position here. And as protons flow, this gamma subunit moves. And you can see this notch has moved from here to here. And that movement allows for the release of ATP from that subunit. And so note that as gamma moves, ATP is released over here. This ADP is converted to ATP, and then this space, which is empty, it binds ADP and inorganic phosphate. And then gamma moves again. So note gamma moves from here to here, and then this ADP to an inorganic phosphate is converted to ATP. This ATP is released, and then this empty space binds ADP and inorganic phosphate. Gamma moves again, and then you see here ADP gets converted to ATP, ATP gets released, and you have binding. There's some really good kinetic information that comes out here, but we won't go into it for this lecture. I might do that in the live class though. Um, but I think just in terms of data interpretation, it's important to go through this one. And so here, this says kind of two things. One, ATP production requires the electron transport chain, right? And that makes sense, right? Because the electron transport chain is making the proton gradient that is allowing for ATP to be produced. So this is something that hopefully everyone understands if we've been kind of keeping up. You need the electron transport chain in order to get ATP production. But what this graph says is that if you block ATP synthase, you also block, so here in black is oxygen consumption, which is a measure of the ETC, the electron transport chain, and then in red is ATP synthase, right? And so by blocking ATP synthase, so oligomycin or venturocidin, I don't know how to say that. Shoot. It blocks the ATP synthase. And when you do that, right, obviously you don't make any more ATP, but you also stop the electron transport chain. So not only is the ATP production dependent on electron transport chain, but the electron transport chain is dependent on ATP, dependent on ATP production. And so one of my second favorite test questions is, why is that the case? And I will leave that to you to answer. And I'll say it's in the data. Um, yeah. And then, I'll actually end up stopping here because I know I've gone long. There's a lot more that goes through it, but we'll talk about that in the live class. Okay, I will see y'all soon.